Hello everybody. Welcome back.
Hello, welcome back to Getting That Medical Degree. Welcome back to our channel where we talk about everything medical related. But this time we're doing something different in today's episode.
Yeah,
What are we doing,
so
Zach?
Mcat, stuff to get you into medical school. So,
All right,
so let's get
take it
into
over, Zach. What are we gonna talk about today
it.
this new format of MCAT Academy?
Yeah, so this is our new series called Mcat Academy. We're starting off with biology because I think that's probably the easiest to ease people into studying for the Mcat. It's probably gonna go biology, biochemistry, general chemistry, then OChem, and
We should
then
do.
hmm?
Yeah, yeah. That
Yeah,
makes
you
sense.
good? But we're just going through, basically this is made by using multiple sources such as the Kaplan books, the Cracker books, and basically anything else I find on the web. So yeah, this is about the cell for the first episode. So the parts of the cell, nucleoid, this has the DNA region in the prokaryotes. The nucleolus makes the ribosomes. This is in the nucleus in the membrane. Peroxisomes is collects and break down the material. Rough and smooth ER, basically smooth, accepts the blah. It makes the detox and stuff while the rough makes the protein. Sorry about that. Golgi modifies and distributes protein. Centrioles, 9 groups of microtubules, pull chromosomes apart. Lysosomes are kind of like the demo and recycling center. They have a single membrane. And then the plasmids are in prokaryotes, and they carry DNA. It's like a little thing prokaryotes have. They can like
Now
share
this
between stuff. Yeah,
Now, you think this would be like one of the most basic things to study on your own besides watching this video, right? So these are like important sections that you need to know
yeah. So like this is made by
and
going through
understand
like
how they work.
yeah. So this is made by going through like 2 different books and like taking out the highest yield stuff and then doing some UWorld questions too. So
Okay.
basically, you have to know what the parts of the cell do, and pretty much every detail in the slide's high yield. I tried not to include anything that's super low yield. So, like knowing the lysosomes of single membrane, it's probably important too. Knowing what the rough ER does versus the smooth ER, it's important. Pretty much everything there is important. Like where the nucleolus is sitting, it's in the nucleus, which is an easy, like, quick question they might ask you on the test. So, yeah. Bacteriotypes. So, obligate aerobe, which means it needs oxygen. Same thing, but it changed to anaerobe, it dies in oxygen. Facultative anaerobe, so that means it can switch between both aerobic or anaerobic. This is pretty easy. And then aerotolerant, it doesn't need oxygen, but it tolerates it. So basically, if you understand the wording of it, you only have to memorize the definitions. So the wording basically tells you what it is. But if you didn't know the wording, you probably could miss on the test. Gram positive versus gram negative bacteria. So gram positive stains purple and has a thick wall, while gram negative is pink and it has a thin wall. So that's important outside of the MCAT in general, just in general medical stuff. That's important for when you're staining bacteria for infectious disease. You know if it's a gram positive infection and gram negative infection. Knowing that just in general can decide what antibiotics you use, and it really cuts down like what you need to use. So if it's something's gram negative, you're not going to use Vanc, for example, because that's a gram positive only antibiotic. So that automatically cuts down your what you're going to use and kind of narrows it down without even knowing what the culture is yet. So some outside knowledge. You get on this slide, Mitchell?
Yeah.
Okay. Then you have eukaryotics versus prokaryotics is just an easy comparison. So ETC, electron transport chain, is mitochondria and eukaryotic, while it's in cell membrane for the prokaryotic. Again, easy things they can switch up on you. Like, oh, this is a prokaryotic, and you pick mitochondria on accident, and you're like, oh wait, that's cell membrane. Large ribosomes versus small ribosomes. Then reproduction is mitosis versus binary fission. Also, prokaryotics carry a plasmid which carries DNA material, as we talked about recently. Prions versus viroids. So viroid is a plant pathogen, while a prion is an infectious protein, so PP. That's kind of how I think of it. This can trigger misfolding, which it calls like alpha helical, change to beta plate sheets, which basically decreases the solubility of the protein stuff. So this gets we get more detail in prions in the biochemistry part, where this is kind of like where you see more of this stuff at. But this is just like a general introduction to it, which is why we're not going into detail like the alpha helical and stuff like that yet. The cytoskeleton, known what it's made up of, you have microtubules, which is actin, micro microfilaments, which is actin, microtubules, which is tubulin. So tubules, tubulin, filaments, actin. There's an A in filaments, so you can think of that as actin. That's kind of how I thought of it. And then intermittent filaments, keratin, which is equals to bimetin, and desmin to lamin. Um, so tissues. Everyone knows in your basic biology class where they have you give you a picture of a tissue and you have to name it. That's the same for the Mcat. So knowing like the names of it, but also being able to see pictures of it is pretty important. So like simple as one layer, stratified as multi, pseudostratified is probably the harder one. It looks like it's multi-layered, but it's actually a single layer. And they combine these, so they would be like a pseudostratified cuboidal one, or like a stratified columnar or squamous. So you have to know like how to describe the shape of it, but also describe like how many layers it has. That's kind of how the MCAT would work on this. Connective tissues is like stroma, which supports the for support extracellular matrix, bone, cartilage, tendon, and blood. Again, this is just like the introduction to biology, pretty much. So this is kind of like big picture looking in, and then we'll go into more detail later on. So, so genetic recombination. This is like so transformation. You get genetic information from the environment. This is like bacteria. And then conjugation is how they transfer genetic information. So it's like a via conjugation bridge. So it usually goes F positive to an F negative, which is the terminology they use on like the MCAT, which I didn't know about for a while. So I'm like, what's this F positive mean? What's that? I'll do some looking into it. So there it is. Transduction is transferred from a bacteriophage. So bacteriophage like gets on top of the bacteria or something, it injects stuff into it. It's kind of an example of that. And then transpons is like when genetic information can insert and remove themselves. So, and since we talked about bacteriophage in this slide, the next slide's about kind of about them. So they have a capsid, not a capside, which is a protein coat around them. Some have a lipid envelope around them. There's a viron, which is virus particles. And the bacteriophage itself, the bacterial virus, has like a tail, and that injects like DNA or RNA into like the cell or whatever it's infecting. And that can be DNA or RNA. And its viral genome can be DNA or RNA or single stranded or double stranded. So SS means single stranded, and DS means double stranded. So single strand can be a positive sense, which can be translated by the host cell, while a negative sense requires like RNA replicase, which must be used to synthesize a complementary strand. I mean, this is like kind of simple concept, but I missed questions on this because I didn't really know the terminology.
Terminology is a killer.
I didn't think my biology class taught that positive strand versus negative strand. I don't know about yours. Did yours, Mitchell?
I do not remember.
Yeah, so I was doing some U-roll questions. I saw that pop up. I'm like, what is that? Like, I have no idea what that is. Hence, here it is. So, so the positive sense can be translated without replicase, while negative strand needs the replicase. And so they also have lytic and lysogenic cycles. So basically, lytic basically replicate inside the cell until the cell lyses. When a cell lyses, like or breaks open or dies, pretty much, that releases like the I guess the bad stuff everywhere else, and now that infects multiple cells, and that makes more cells lyse, and it keeps spreading around the body. Well, lysogenic, where it just sneaks itself into like the genetic genome, and it keeps replicating, and then it like just deactivates until something else reactivates it later on. Knowing those two things is probably important from the slide, also known positive sense and negative sense. And then cell cycle. So G1, you make mRNA and proteins to prep for mitosis. G0, so this is where the cell goes if it does not need to divide. There's checkpoints. So G1 checkpoints is after G1. Okay. Does the cell need to divide? Or should it divide? Okay. It should divide. Now it goes to S. P53 is in charge. Not 100% sure how detailed the Mcat will go with P53. P53 itself is really detailed. If you go in cell biology, we're having an entire like lecture on just P53 and like all its other stuff that goes with it. So the books itself didn't go in detail on it, so I'm not going in detail right now. But just know that can be pretty detailed. So then you go to S phase, DNA replicate is replicated. Then you go to G2. The cell grows. It makes the parts of the cells, so which is called organelles. You get to a checkpoint. So how's the cell size doing? Does it have the right amount of organelles? Everything good? And you get mitosis and cytokinesis. Then you have positive growth signals and negative growth signals. So positives, CDK plus cyclin creates complex. It's phosphorylated RB to RB plus P. The P is like phosphorus. And RB changes the shape, releases E2F, and cell divisions continues. Then you have negative signals. So CDK inhibits the phosphorylation of RB, so the E2F stays attached and the cell cycle halts. So that's really about it for the cell. So
It's quite a lot of information, so it's probably gonna take, depending on how fast you digest information, probably a week. I am trying to slow down to how fast I digest information, and when I'm trying to like go really deep into this.
it is a lot of information, especially if this is your first time seeing it. If like you're like an accounting major or something, like this is a lot to digest in like 11 minutes.
Yes,
So
yes, it is quite a bit. But these are good headlights. If we miss something, please put it in the comment down below, and we will add another revised Cat Academy bio with extra stuff
yeah, so
in the future.
stay tuned for the next video in the series. Peace.